Yeah.
Okay.
The first two chapters, right?
Yeah, yeah.
Yeah, okay.
Okay, then you will be able to answer if you have revised it nicely.
Yeah.
Okay, great. So right now we're going to talk about aerobic respiration. Now earlier we saw that there were three fates of the pyruvic acid which was formed after glycolysis. The first one was alcoholic fermentation, then there was lactic acid fermentation, and the third one was aerobic respiration. The first two scenarios were for the anaerobic condition, whereas the last is the aerobic condition, right? Meaning here oxygen is present. Okay. So talking about the aerobic respiration. So basically here what happens is that a complete oxidation of glucose takes place and then energy is stored to synthesize larger molecules of ATP or larger numbers of ATP. Okay. So if we are talking about the eukaryotic organism, so the process of aerobic respiration takes place in the mitochondria, and obviously since it is aerobic, it would require oxygen. This much is clear? Hmm?
Yes.
All right. So talking about the aerobic respiration, so it is the process. It is the process by which organisms can carry out the complete oxidation. Do you remember that in the fermentation process, incomplete oxidation or incomplete breakdown was being done?
Yes.
Yeah, but here the complete oxidation will be taking place. But oxidation of what? So, complete oxidation of glucose molecule will take place. Okay, and
Yeah.
we'll extract the energy like when the oxidation is being done, so we'll extract the energy which is stored, so we are extracting the energy to synthesize larger numbers of ATP, larger number of ATP. Okay.
Yeah.
Yeah. Now let us look at the site of aerobic respiration. So in eukaryotes, in the case of eukaryotes, the site of aerobic respiration, site of aerobic respiration is the mitochondria. Okay, mitochondria. Always remember this one thing because this is asked in the MCQs also. Where what is the site? So the site is mitochondria. Now, what happens that during the process of aerobic respiration, it takes place first of all in the presence of oxygen and ultimately carbon dioxide, water, and a large amount of energy is released. Okay, so the aerobic respiration takes place in the presence of oxygen and releases carbon dioxide, water, and a large amount of energy. Is it clear up till now?
Yes. You
Hmm.
weren't on your camera today.
What?
You won't sit on your camera today.
Oh, I'm so sorry. I completely forgot. All right, switch on your video. I forgot. All right, am I visible?
Yes, I can see.
Okay, okay. Now see. So obviously during the process of aerobic respiration, we'd require oxygen first of all, and then this oxygen would be used, and the oxidation of the substrate which will be taking place, and ultimately we'll have the release of carbon dioxide, water, and a huge amount of energy. Got this much is clear, right? All right, now see, the process of aerobic respiration actually happens with a lot of intricate things or intricate reactions that do take place inside it. Okay,
What is
so
intricate?
something complicated happening inside. Okay, so basically for like the whole process of aerobic respiration, first of all, it takes place in the mitochondria, right? Now the pyruvic acid which was formed at the end of glycolysis. Okay, first of all, you tell me the process of glycolysis takes place in which part?
Cyto.
Hmm. Cytoplasm, yes. So after the pyruvic acid is made in the cytoplasm, from the cytoplasm it is being transported to the mitochondria.
Yeah.
Because we are talking about the fate of pyruvic acid,
Yeah.
right? So from the cytoplasm, the pyruvic acid after the glycolysis comes into the mitochondria. Then in the mitochondria, the whole aerobic respiration events start. So first of all, what happens is that complete oxidation of pyruvate happens, and during that process, stepwise removal of all the hydrogen atoms take place. Meaning from the
So,
pyruvate,
what about the formation of lactic acid and ethanol and carbon dioxide?
that is, see, the three things that I said, all these three things are different, independent of each other. Okay, but glycolysis is that one common thing which is happening in all of them. Why? Because at the end of glycolysis, we have pyruvic acid made, right? Now, that pyruvic acid can either undergo alcoholic fermentation. This does not take place in our body, right?
Okay.
Alcohol do not
Yeah,
make
yeah,
alcohol. And the second way by which the pyruvic acid can be acted upon is when it will undergo lactic acid fermentation. Now, this
yeah.
can take place in our body when we are doing exercise or during the lack of oxygen. But for aerobic respiration, now in our body, in our cytoplasm, when glycolysis has been done, pyruvic acid is formed. Now, if we run or do anything of that sort, then this pyruvic acid will turn into lactic acid. Hmm? But under normal conditions wherein we need aerobic respiration to take place, so in that what will happen? From the cytoplasm, the pyruvic acid will be transported out and it will go into the mitochondria. Then only
Okay.
aerobic respiration.
Yeah.
Okay. Now after the this thing, pyruvic acid has been transferred from the mitochondria, sorry, from the cytoplasm into the mitochondria, then oxidation of the pyruvic acid takes place. Now, oxidation can be said as adding of oxygen or
Yeah.
removal of hydrogen, right?
Yes.
Of them are
Yes.
oxidation.
Oxidation.
But yes, during that process, oxidation of pyruvic acid takes place, and in that oxidation process, stepwise removal of hydrogen atom takes place. Again, that is oxidation only. Hmm?
Of pyruvic acid.
Yeah, hydrogen atoms are being stepwise
Okay.
removing. It's like we are removing it, and at the end, we just have three molecules of carbon dioxide. Meaning, when all the molecules of hydrogen atoms have been removed, and that process is oxidation, right? So at the end, we are left with three molecules of carbon dioxide, CO2. Now, later on, what will happen? First of all, um, from this CO2, we get it converted into acetyl coenzyme A. This is a compound. Okay, so
From where
listen.
did CO2 come from?
Hmm?
From where did CO2 come from?
You took pyruvic acid. You removed all the hydrogen which was there.
Yeah.
What we are left with is three carbon dioxide.
Oh, you said.
CO into three molecules of CO2.
Oh, I thought you said three carbon atoms.
No, all the hydrogen atoms are removed. Carbon
Okay.
dioxide. Okay,
Okay.
and after that, this acetyl, sorry, these three carbon dioxide atoms, they are not atoms, molecules, they are converted into acetyl coenzyme A. That is the first step of aerobic respiration. Okay, conversion of pyruvic acid into acetyl coenzyme A. Okay, later on, the tricarboxylic acid cycle, that is Krebs cycle, takes place. Okay, that is the second step of the aerobic respiration. So first, acetyl coenzyme formation, then TCA, which is tricarboxylic acid cycle. Okay, this much is clear?
Yes.
So we'll write down first of all. All right, yes. So we'll write that the pyruvic acid formed in the cytoplasm by the process of glycolysis gets transported from the cytoplasm to the mitochondria. Now, if somebody asks you, like, why is the pyruvic acid transported? Now you can answer. Why is it transported?
For aerobic.
Because aerobic respiration takes place
Yeah.
in the mitochondria.
Yeah.
Okay.
Yes.
So first that happens. Now, the first thing that takes place is that the complete oxidation of pyruvic acid.
Your voice is kind of low.
Is it fine now?
Yeah.
So the complete oxidation of pyruvic acid takes place by the stepwise removal
Mm. Hydrogen.
of the hydrogen atoms. Okay. So at the end, how many of what molecules are left?
3 carbon dioxide molecules are left.
3 CO2 molecules are left. Okay. Now, the first thing that
And
happens,
then carbon dioxide is converted into acetyl
coenzyme A.
coenzyme A.
So first thing that happens after the like with this carbon dioxide is that formation of acetyl coenzyme A. Just 1 second. So yes. All right. So first of all, the first thing that happens is that formation of acetyl coenzyme A takes place. Excuse me for 1 second. Okay, so after this process, what happens is tricarboxylic acid cycle, which is the TCA. Okay, so tri acid T cycle. Okay, this cycle takes place. Tricarboxylic acid cycle. This is TCA. Okay, tricarboxylic acid cycle. Yes.
First, the carbon dioxide molecules are converted into acetyl coenzyme A, right?
Hmm. From CO2. I'm sorry, I was distracted. So we'll write from CO2 over here. Okay. And then later on, the tricarboxylic acid cycle takes place, and you can also call this as Krebs cycle. Krebs cycle, or you can also call it as citric acid cycle. These are the names. All of them mean the same thing. Okay.
Okay.
Now, later on, see, do you remember that the mitochondria had a matrix?
Yeah.
Hmm? Do you or do you not? Mitochondrial
I remember.
matrix, crista, you remember all those things? The
Yeah.
infoldings and everything? Yes.
Yeah.
So the process, which is TCA, okay, it takes place in the mitochondrial matrix. Like how the cell has a cytoplasm, the mitochondria has the mitochondrial matrix. So this process of TCA takes place in the mitochondrial matrix. Okay, and later on, wait, who left?
Learnivio left.
Hmm. The host left, the co-host left. Okay, maybe some student left. Khadija did not join today. All right. So later on, yes. So this process takes place in the same. What happened? All right. Yeah. So this takes place in the mitochondrial matrix. Now later on, what happens is that all the electrons that were formed over here, or or the electrons that were removed as a part of hydrogen atoms, okay, they go through the ETF, that is electron transport system. But that is for something later on. First, we'll look at this particular thing. Okay. Now, talking about the first step, that is the formation of acetyl coenzyme A. Okay. Now, let us see how does that take place. So, I don't know what is wrong with this screen today. Hmm. So, the first thing that we have to see over here is the
Change the
formation
color.
in the color. Galaxy again. Oh, sorry, you were choosing.
Yeah.
Hmm. So Teja, you feel free to choose the colors because apparently we are all maniacs. So let us choose the color. You can also suggest a color, okay? Things
Yeah.
that I do.
Take the
Hmm.
blue one.
Which
The
one?
third,
This?
third, the
Ew, it's
no,
ugly. Which one?
the third line, the from the left, the first one. Yeah,
This?
that one. Yeah.
For the heading, I'll choose it for the body.
Oh, okay, okay. For the heading, choose the last line one.
This? Shall I do a rainbow?
Yeah, yeah.
You're fine with it, Khudeja?
Yeah, yeah, yeah. Oh,
Yeah, mom.
Everyone? Yeah, everyone should chip in the madness. All right. So first of all, we're going to see the oh, it's cool. Formation
wow.
of acetyl coenzyme A. And this whole process of the formation of acetyl coenzyme A is also known as oxidative oxidative decarboxylation. Decarboxylation. So oxidative decarboxylation is the process, or you can also call it as the formation of acetyl coenzyme A. Okay. Now,
Yeah.
let us see what actually happens in this particular process. So see, the pyruvic acid, which is formed by the glycolysis, right? So the that process took place in the cytoplasm, isn't it? Now, as soon as the pyruvic acid is transported into the mitochondria, then what happens? It gets acted upon by the enzyme that is known as pyruvate dehydrogenase. Okay, pyruvate dehydrogenase. And with that being said, because of this enzyme and also in the presence of magnesium ions, the pyruvic acid gets converted into acetyl coenzyme A. Okay,
What acid?
pyruvic acid, the end product of glycolysis. Okay, so
This is after the removal of hydrogen atoms.
no, this is much, much before that.
Oh,
Meaning, listen, like as I said, that pyruvate dehydrogenase is there. Dehydrogenase word you understanding?
yeah,
Dehydrogen,
removal of
right? Removal of
hydrogen.
hydrogen. Yes. So those when those hydrogen atoms are removed, along with that, the pyruvate gets turned into the acetyl coenzyme A, and then we have this thing also, carbon dioxide production side by side in the product. You'll see the reaction and then you'll understand what I'm trying to say. So basically, the pyruvic acid gets acted upon gets acted upon by the enzyme which is known as pyruvate pyruvate dehydrogenase. Okay, pyruvate dehydrogenase. Now, when this when this takes place in like during this particular process, what happens is that the pyruvic acid gets converted into acetyl coenzyme A. So if we write the reaction, so we'll write pyruvic acid, which is a 3-carbon molecule, right? Now this pyruvic acid, and we also add coenzyme A as a substrate only, okay? And then we add NAD plus, meaning the redox equivalent that is added. Then in the presence of magnesium dipositive ions and pyruvate dehydrogenase, okay, in the presence of this enzyme, what do we get? We get acetyl coenzyme A. This is how you write it in short. And this is a 2-carbon molecule. And along with that, we also get carbon dioxide. Then since we have added NAD plus, so we'll also get NADH plus H plus. This is the whole reaction of oxidative decarboxylation. Okay. This much is clear.
Yeah.
Any doubts? Also, look, these CoA, NAD plus, these are coenzymes. Now, do you remember what coenzymes are? Coenzymes, prosthetic groups, metal
Yeah.
ions. Remember?
Yeah.
They were added to the reaction, right? So that
Yeah.
it can increase the rate of the reaction. Same way for this process, coenzyme A, NAD plus, both of them are used as certain coenzymes so that this reaction can happen at a faster rate. Hmm? This much is clear?
So pyruvic acid from cytoplasm it comes to mitochondria, and then after that,
After that, this pyruvic acid which was transported, it gets converted into acetyl coenzyme A by the action of pyruvate dehydrogenase. Okay,
so the hydrogen atoms did not get removed yet.
they did. They did.
You said it did happen before.
No, see, in this particular step, now you see there is no trace of pyruvic acid anywhere in the reactant side. This pyruvic acid got converted into acetyl coenzyme A
Yeah.
and then CO2 was left. And how did I say that 2 molecules of CO2 will be left? How did I say that? Or how did I say that 3 molecules of CO2 will be left? Just
After
because,
hydrogen atom
yeah.
split.
So, okay, first you tell me one glucose molecule forms how many pyruvic acid? Hmm?
One,
One glucose molecule after undergoing glycolysis forms how many molecules of pyruvic?
two,
Two
two.
molecules, right? So one pyruvic acid will make one CO2 at the end, then two pyruvic acids will make two CO2 at the end. Getting my point? Also, this process, or you can say the formation of acetyl coenzyme A or the oxidative decarboxylation, this process, it is also known as link reaction. Okay, why?
This thing,
Yeah, this whole thing. It is a link reaction. Why? Because it is sort of acting like a link between the pyruvic acid and the TCA, which is the tricarboxylic acid cycle. Because in order to enter the TCA, acetyl coenzyme A is very important. So first acetyl coenzyme A must be formed and then only it can enter the TCA, right?
yeah.
Yes. So that is how the whole process is done. All right. So after that you write down two molecules, two molecules of pyruvic acid. You write down the full. Formed or are formed, two molecules of pyruvic acid formed from one molecule of glucose, one molecule of glucose during glycolysis undergoes the formation of two molecules undergoes the formation of two molecules of acetyl coenzyme A, two molecules of CO2 and two molecules of NADH plus two molecules of H plus. So these are the things that are formed. Also, this reaction is called as the link reaction. As the link reaction, or you can also call it as the gateway reaction. Gateway reaction of aerobic respiration. Got it? This much is clear?
Yes.
Okay. Now, since this process is done, now we'll move on to,
Wait,
you want,
let me
okay,
write.
okay. Now we'll move on to the TCA, that is tricarboxylic acid cycle. This is the link reaction, meaning it is actually connecting the pyruvic acid to the TCA.
Done.
Now, before you mention it, wait, let me try something else. Then we'll write TCA cycle. Okay, now let us see what happens in this particular cycle. So, now the acetyl coenzyme A, which was formed at the end of the oxidative decarboxylation, right? After that process, the acetyl coenzyme A enters a cyclic pathway, and this cyclic pathway is known as Krebs cycle. And obviously, it would be named after some scientist. So his name was Hans Krebs. Okay, so this cycle is named after him because he was the first person to ever talk about this thing. Actually, he saw or he observed it in the flight muscles of pigeon. I don't know how, but he did. So because of that, this cycle is named after him. So it is Krebs cycle named after Hans Krebs. Okay. Now what happens? From the acetyl coenzyme A, in this particular cycle, stepwise hydrogen atoms are removed. Okay, so in the TCA, stepwise removal of hydrogen atoms from acetyl coenzyme A takes place, and then those removed hydrogen atoms are transferred to the coenzymes for further processing in the ETS. We'll have to study the whole ETS. You remember in photosystem 1 and photosystem 2, ETS was there in the middle.
Yeah.
That ETS we have to study about it. Okay, but that's not the point of concern right now. The point of concern is TCA or the Krebs cycle. Okay, so the Krebs cycle, now the hydrogen atoms which were removed, that they are going to the ETS, and the acetyl coenzyme A, it gets changed into certain components or certain compounds throughout the whole cycle. Okay, so let us write down something about the TCA. So first of all, the acetyl coenzyme A acetyl coenzyme A enters a cyclic pathway called as the Krebs cycle. It was named after Hans Krebs, who first elucidated it. Okay, so wait. Now in these reactions, as I said, in these reactions, the hydrogen atoms, the hydrogen atoms are removed from the acetyl coenzyme A. From the acetyl coenzyme A. Okay, and and they are transferred. You should mention it also. So, and they are transferred to the ETS. Okay. Now, see, the cycle, as I told you, that the TCA or the Krebs cycle is also named as citric acid cycle. But why? So there is a reason, because like this particular cycle is also known as citric acid cycle because the first stable product is citric acid. Okay, so that is why the name. So we'll also mention that the cycle is also called as citric acid cycle, as citric acid is the first, or you can say is the first formed compound, is the first formed compound.
We can't say stable.
No, you shouldn't, because you know it soon changes its whole entire thing. Okay, it gets changed. So it is the first formed compound of the cycle. So that is why the cycle is also known as citric acid cycle. Also, it is known as tricarboxylic acid cycle because many intermediate compounds, they have three carboxyl groups. Okay, that is why. Like there is a reason behind having a different kind of name. Okay, so this was the reason for having
What is intermediate compound?
something that is formed in between. Like one is
Oh,
the end result, and something which is like all the compounds that are formed in between getting to the end result, those are intermediates. Okay, so uh, it is also called as TCA. You write down the full thing. Okay, tricarboxylic acid cycle, TCA cycle, because many many intermediate compounds many intermediate compounds formed in this cycle in the cycle have three carboxyl group. Carboxyl group, you understand? You might have,
carboxylic acid.
yeah, that is what? How do you write it in
C
chemistry?
COOH.
Very good. So it has three carboxyl groups. Got it? Why is it known as citric acid cycle? Why is it known as TCA?
Yes. Yes.
All right. Okay. Very good. Now moving forward, see, in this particular cycle, oxygen presence is very important, isn't it? And this whole cycle takes place in the mitochondrial matrix. Now, this cycle is a common oxidative pathway for carbohydrates, fats, and proteins. So all of this would undergo TCA. Okay, carbohydrates also undergo TCA, fats and proteins, they also undergo this particular cycle. Okay, now all the enzymes that are used in this particular cycle, okay, they are soluble in the mitochondrial matrix, meaning they are present, like they can be transferred into the mitochondrial matrix. Okay, but there is an like exception over here, is that there is an enzyme which is known as succinate dehydrogenase. Okay, SDH. This is found attached to the inner mitochondrial membrane. Do you remember that mitochondria is a two membrane structure,
Yes.
double membrane structure? One was the outer membrane, one was the inner membrane. Now, the inner membrane encloses the mitochondrial matrix. You remember? Outer membrane, and then there was an inner membrane that had convolution all over, right? And this part that it enclosed was the matrix. So all the enzymes that are used in this particular cycle, they are soluble in the matrix except for one enzyme, that is succinate dehydrogenase, and it is present in the inner membrane here. It cannot come here because it is not soluble in the mitochondrial matrix. Got it? This is important. This line that I am saying or explaining right now, it is important. So all the enzymes are soluble in the mitochondrial matrix, but succinate dehydrogenase, okay, succinate dehydrogenase, that is SDH, is found found attached to the mitochondrial membrane inner mitochondrial membrane. This much is clear to you?
Yes.
Any doubts up till now?
No.
Okay, very good. Now moving forward, we will look what happens in this particular cycle to see. During this, it starts with the condensation of the acetyl group with oxaloacetic acid. Okay, so the acetyl coenzyme A, it gets condenses, or you can say it condenses with the OAA, that is oxaloacetic acid, and then it forms the citric acid cycle. Just wait till I, actually add the flow chart of the cycle, then it will be better to understand. Just wait for 1 second. Okay, are you people able to see the image that I added? Is it visible?
Yes.
Let me just find it. Okay, now it's visible properly.
Yeah. That's from your book.
Yeah, it's from my own book. All right. Now see, we saw that this pyruvic acid is being converted into acetyl coenzyme A, right? This much is clear, right? This whole thing, this part. This is known as the oxidative decarboxylation. See, I've written wrong. Oxidative decarboxylation, right? Now, I am saying that this particular acetyl coenzyme A, it condenses with the oxaloacetic acid. So oxaloacetic acid comes and mixes with the acetyl coenzyme A, and then we have the citric acid, which is the first compound formed. This much is clear? No? What happened? What is the doubt? Tell me.
What is condenses with ox?
See, because the reaction takes place in this way. There is no reason why it happens. It just happens.
No,
You need,
what is the meaning of condenses?
see, basically, it actually mixes. Just imagine it like mixing it with the acetyl coenzyme. Okay. Hmm?
So there are two things happening.
Tell me what are they?
It condenses with oxaloacetic acid and
Hmm.
citric acid.
No, it condenses with oxaloacetic acid and forms the citric acid.
Oh. So first this happens and then that happens.
Hmm?
Okay, I understood.
What happened?
Nothing.
Okay. See, yes. So basically, what is happening is that condensation of the acetyl coenzyme A along with the oxaloacetic acid is taking place, and because of this, we are getting citric acid. Meaning, citric acid is being formed. Getting my point? So here citric acid is being formed, and the enzyme used for this reaction, for this condensation reaction, is citrate synthase. This is the enzyme, citrate synthase. And during this process, the coenzyme A molecule is released. Got it up till now? Shall I clean the diagram so that you can understand it better? Okay. This is combining with this to form this. Now it's clear. And during this process, the enzyme that is acting is the citrate synthase, and the coenzyme A is getting released. Okay. Meaning it's not needed anymore. Now later on what happens is that this citric acid, it gets isomerized to isocitrate. Okay, see, these are like, there are so many steps, but we do not have to know each of them because NCERT does not mention it. Do you remember like how in glycolysis, the glucose 6-phosphate was being isomerized into fructose 6-phosphate?
Yes, yes, yes.
Same way, the citric acid, or you can also call citric acid as citrate. Okay, so this citrate gets isomerized into isocitrate, from citrate to isocitrate. Okay.
Okay.
Now, later on, the
The diagram in my book is different.
the diagram in your book is different.
Yeah.
Yeah, see, the NCERT one.
Hmm.
Yes, it will, because they haven't shown all these steps, all the real steps that do take place. They have cut out many steps. They have
Oh,
mentioned isocitrate being formed, but they haven't shown it being formed in the chart, right? They
yeah,
haven't
yeah.
shown it. Yes. So that is why I'm using this so that you can understand that, yes, okay, isocitrate, here it is being formed. Okay. Now later on, the isocitrate undergoes 2 successive steps of decarboxylation, meaning the removal of carbon dioxide, right? So
Hmm.
decarboxylation happens, and then we have alpha ketoglutaric acid. This is made. Okay. Alpha ketoglutaric acid is made, and then by the next dehydrogenase complex, succinyl coenzyme is made.
Wait,
What?
I'm still processing.
The isocitrate, I'm saying it again, okay? The isocitrate went under successive decarboxylation, okay? And then after the this thing went under decarboxylation, the first molecule formed was alpha ketoglutaric acid, and then again, then there was succinyl coenzyme was formed.
What is oxalosuccinic acid?
Again, not mentioned, does not need to be mentioned. Okay,
Okay, so
see,
we
the
will say
compound that I,
isocitrate.
yes,
Isocitrate goes through successive decarboxylation and
yes,
then alpha ketoglutaric acid is
acid,
made.
yes. And
Okay.
like for like after first decarboxylation, alpha ketoglutaric acid is made. Then after
Okay.
second decarboxylation, succinyl coenzyme A is made.
Okay.
And remember one thing, that the process of decarboxylation is basically the removal of carboxyl group, okay, and the release of CO2, isn't it? That is
Okay.
what happens. So
Yeah.
basically, after that, like after the succinyl coenzyme A is formed. Okay, this much is clear.
Yes.
Now, after that, what happens is that the succinyl coenzyme A, it goes through various steps, ultimately to regenerate or form the oxaloacetic acid again. Again, these steps are not mentioned in the book, but I will tell you. Okay, so no, you should know. You should know how the compound is
I
being
get
formed.
confused.
All right. So see, ultimately, oxaloacetic acid is just like regeneration. meaning the cycle will keep on going if the
Yeah.
oxaloacetic acid was there. Now, during the conversion of succinyl coenzyme A to succinic acid, meaning from here to here, GTP is synthesized. Like how ATP is there, adenosine triphosphate, GTP is there, just like an intermediate, energy intermediate, it is guanosine triphosphate. Do you remember adenine, guanine?
Yeah.
Right? So when that adenine was added with TP, then it was ATP, right? Adenosine triphosphate. Now, if you
Yeah.
replace adenine with guanine, then you'll have GT. That is guanosine triphosphate. Okay. So during the conversion of succinyl coenzyme A into the succinic acid, GTP is synthesized. And again, since an energy intermediate is being synthesized, so we can call this particular step, meaning the conversion of succinyl coenzyme A to succinic acid. This particular step can be called as substrate level phosphorylation. Try to recall glycolysis.
Yeah, because GTP is made.
Because we are making the GTP from the intermediate compounds, right? We are using
Yeah.
these compounds as the substrate. This is important to remember. Okay. Now later on, what would happen is that so this when this GTP is converted into GDP, okay, it is just like how ATP is converted into ADP, meaning whenever energy is used, it is just like that only. Getting my point? Now see, if you look at this particular step, you'll find that FAD is being converted into FADH2 and here NAD is being converted into NADH2. Like how NADH2 and ATP are the energy intermediates, same way FADH2 and GTP are also energy intermediates. Okay. And where else is this NADH2 being synthesized in the cycle? Here, right? From
Really?
NAD to NADH2,
Oh, okay. Okay.
right? Meaning during the
Yeah.
second decarboxylation. So we can see in the whole Krebs cycle, there are 3 points where NAD is reduced to NADH2, isn't it? Hmm?
Mm.
Meaning one, this is the first point, this is the second point, and this is the third point. 3 places where NADH2 is being formed, and then one
Yes.
place where FADH2 is being formed, isn't it?
Yes.
Yes. So basically, these are all energy intermediates and they are extremely important for the whole process to occur. You've understood the Krebs cycle?
Yes.
Clear?
Can I can I say it in my own words once?
Hmm. Say it.
So acetyl coenzyme A,
Hmm.
it condenses with oxalic acid.
Oxaloacetic acid.
Oh, oxaloacetic acid, and then citric acid is made,
Hmm.
and then uh, the citric acid isomerizes,
Very good.
and
Two.
then it becomes isocitrate,
Correct.
and then after that, um, the first NAD is converted into NADH is synthesized.
NADH2 is synthesized.
NADH2 is synthesized, and then uh, the isocitrate, it goes through decarboxylation, the
Very good.
first one, and then it becomes alpha ketoglutaric acid, and
Hmm.
then the second decarboxylation happens, and then uh,
It forms
it forms succinyl
coenzyme A.
coenzyme A, and then also NADH2 is synthesized again the second time,
Very good.
And then from succinyl coenzyme A, succinic acid is made. Over here, GTP is synthesized.
Mm.
And then after that, the regeneration happens. And
Mm.
then oxaloacetic acid is made again, where NADH2 is synthesized again. Third time.
Got it? Exactly.
Yeah.
This is how the whole cycle is. Okay.
Okay.
Now, if you want to look at the reaction for this particular cycle, so we'd say pyruvate. It's a little long reaction. So pyruvate acid plus 4NAD plus plus
What is this?
wait FAD plus. You'll see it. Then 2H2O. This is actually sort of like a conclusion for this whole cycle because see H2O is here. We didn't count it, right? So we are now going to do it. So, okay. So this pyruvic acid plus 4NAD plus FAD plus 2H2O plus ADP plus PI, that is inorganic phosphate, gives us in like it happens in the mitochondrial matrix. Now what happens here at the end? We have 3 molecules of CO2 plus we have 4NADH, right? Plus 4H plus we have FADH2 and then we also have ATP. This is the summarized version of this whole entire reaction.
Which reaction?
I mean this cycle. This reaction is for this cycle.
Why is it different?
How is it different? See, basically, the reason why we are adding 4 NADH, but because before this also, when the pyruvic acid was converted into acetyl coenzyme A, during that process, NADH2 was required. So that is why we are adding it, and this is the whole entire summary, you can say, of the aerobic respiration. Okay? Even if you don't understand it that well, just learn it because they can ask you to write down the equation.
What will they ask us? What is it like?
What is the
The
reaction for aerobic respiration?
aerobic.
Yeah. Is it clear up till now?
Yeah.
All right. Now this was the TCA and it has been completed. Now let us move forward and talk about the electron transport system, which is the ETS. Again, let's make it fun so that you people do not despise this chapter. So, electron transport,
This is rainbow. Galaxy.
yeah, galaxy actually. Electron transport system. We also call it as ETS, not BTS, but ETS. Okay. So the only TS you need to remember is ETS and not the BTS. So ETS is the reaction's name. And later on, what happens is that oxidative phosphorylation also takes place. But before that, we must know about the ETS first, and then only we can move on to the next. So, are you ready for it?
Can you switch on your camera, please?
Oh, my face helps.
Yeah, it's so hard for me to focus.
Okay, wait. The people keep coming around. That is why I'm switching off the camera because it gets recorded and it looks very bad in the recording. That is why I'm
You can blur it there.
It's blur only. My background is blurred. All right. So we saw that whatever hydrogen and everything that was being removed from everywhere, it was being sent to the ETS, right? And ETS is the electron transport system. Okay. Now, do you people know the full form of BTS? Because I don't.
Yeah.
What is it?
Bangtan boys. No. Ah, no. No. I don't know what it is,
I don't know, but anyway, I never wondered. But now that I'm looking at ETS, I can't help but imagine, okay, what is the full form of BTS? Anyway, it doesn't
but
matter.
I am searching.
Okay, you search for it then.
Oh, I was correct.
What is it?
Bangtan, boys.
Bangtan.
Yeah, I think it's
Bangtan boys.
Korean. Yeah, I think it's Korean.
Okay.
Lama, even I don't know.
All right. So talking about the ETS, that is electron transport system. Now, see, in the process, we see, like, basically we saw that the whole glucose was being broken down, certain energy intermediates were being created, and all those things were happening, right? Now, uh, basically in this particular transport system, we are going to see how the electron is actually circulated and it is passed on from one electron carrier. Do you remember electron carriers? Photosystem 1, photosystem 2. Electrons were accepted by electrons, acceptors, electron carriers, such a, yeah. So here the electrons would be passed on from one electron carrier to another electron carrier, and this happens through a series of system, and that is the electron transport system. Okay. So this process, again, the location, it occurs in the inner mitochondrial membrane. Okay, inner mitochondrial membrane. Now see, I will explain it in proper depth only. Okay, then only you can understand as to see, because for TCA, I went along with the NCRT and the way it says. It is very vague, does not explain it in a nice way. But I will explain it in a nice way, the ETS. Okay? Ready?
Yes.
So first of all, what is ETS? So we already know when the metabolic pathway. This is also a metabolic pathway. Okay? So it is sort of like a metabolic pathway.
What
Metabolic
is
pathway,
metabolic?
metabolism, the pathway that takes place inside the body.
Okay.
Hmm? Like, so during this, what happens? The electrons are passed on from one carrier to another. Okay, and this whole process is electron transport system. We are going to see how the electrons are transported. Now be aware that there are going to be many new words over here. So slowly we'll learn each of them. Okay, so let us write that the metabolic pathway through which the electrons passes from one carrier to another is called as the electron transport system. And this is your ETS. Also, talking about the location, so it takes place in the inner mitochondrial membrane. So in the inner mitochondrial membrane. Got it?
Yes. Do you refer a book for making
I
us?
use the NCRT.
So these are your own words.
Yeah.
Uh,
And sometimes I do take the help of certain books also to give you the proper notes, because not everything can come from here. Sometimes I also need help, like certain reference books are there. Okay.
oh,
This much is clear. The location and what happens. Okay.
yes.
Any doubts up till now?
No.
Okay. Now let us see what are the steps of ETS. Okay. Now, the first step. Now see, the electrons from the NADH, which is produced during the citric acid cycle, and where? So in the mitochondrial matrix. So the electrons from the NADH, they are oxidized.
3 times, right?
Hmm?
NADH2 was synthesized 3 times, right? Okay.
So the electrons that are actually produced from NADH being synthesized, so those electrons, they are oxidized by NADH dehydrogenase. Now see, but before that I should make you write the name of the complexes. See, before steps you remove that. Okay, first you write down the complexes that are used. Okay, so complexes used in the ETS. Okay, this whole process of ETS takes place with the help of certain complexes. Okay, so we have complex 1. The name of this complex is NADH dehydrogenase. Okay, this is complex. Okay, this is the
What are
first complex.
complexes?
See, complexes are certain, just imagine them as to being like an apparatus that is like, for example, an enzyme. Okay, the enzyme because of which oxidation is being done, or maybe an enzyme because of which carrying is being done, or again oxidation. So these are oxidizing complexes. Getting my point?
Okay. Okay.
So the first one was NADH dehydrogenase. Then there is complex 2, and this is known as FADH2. Wait, just give me 1 second. Yes. So, yeah, where was I? Yes. So, complex 1 is NADH dehydrogenase. The complex 2 is succinate dehydrogenase. Okay. This is complex 2. Now, later on, there is the complex 3. Complex 3. And these are cytochrome BC1. Complex 3 is cytochrome BC1. Okay, and then at the end we have complex 4, that is cytochrome C oxidase. Cytochrome C oxidase. These are the names. Nothing to be explained, just the names are there. Okay, like how certain people have certain names, same way these complexes have weird names. Got it? Okay, now let us look at the step number 1. Okay, now we write steps of the ETS. Now, because you know now that there are certain complexes which are involved in the process of electron transport. So now let us look at the steps and how those complexes are used. So electrons, the first line, as I said, that electrons from NADH produced in the mitochondrial matrix in the mitochondrial matrix during the TCA, right? Or the citric acid cycle. During the citric acid cycle. I was thinking I'll show you sort of like an animated video for this so that it'll be easier to understand. But after I explain, then only I'm going to show. Okay, so that it'll be like two explanations for the same topic. Okay. All right. So the citric acid cycle uh, are oxidized are oxidized by an NADH dehydrogenase, meaning this is hydrogenase. This is complex number 1. Okay. So here complex number 1 is acting and it is oxidizing. Okay, it is oxidizing the electrons that are made from NADH. Okay, so these electrons from the NADH are being transferred to the NADH dehydrogenase, and then these electrons are being oxidized, or removal of hydrogen or addition of oxygen is the same thing, right? It is oxidation. Okay.
NADH dehydrogenase oxidizes the NADH produced in the mitochondrial matrix.
Meaning
Okay.
the oxidation of electrons is taking place, and when that
Hmm.
takes place, removal of hydrogen takes place, right? So
Yes,
this is the
yes.
first step that happens over here. Now later on, what would happen? The electrons are transferred to ubiquinone. Now ubiquinone is sort of like a molecule which is located within the inner membrane. Okay, it is located within the inner membrane. Now see, as far as I think, because we have studied one more stepwise reaction only, you people will not understand it clearly if I explain. Like, is your mind condensed? Like, have you reached the saturation point that you're not able to get what I'm saying?
No,
Like,
no,
no?
no, I get it. No, I get it.
Okay. What about you, Hudeja? I have done this chapter with Hudeja, right? You remember Hudeja? We did an extra
Yeah.
class on this.
Yes.
Yeah, so this is sort of like a revision for you, right? I hope.
Yeah.
Okay, great. All right, then I'll keep on. Sometimes I feel very guilty that, oh, I'm burdening you people.
What did you do?
Hmm?
What did you do?
I can't hear you.
What did you do?
No, I feel guilty because sometimes I feel like I am burdening, like overburdening you, but then I remember that, okay, that's in your syllabus only. But, you know, I feel
That's
like, okay,
your, that's your
one.
job.
All right, so this was the first step. The first step is clear to you all?
Yes.
Now, talking about the second step, so in this particular step, whatever the electrons are there, so the electrons are then transferred, meaning after the first step is done, then the electrons are transferred to ubiquinone. Ubiquinone. Now, this is sort of like an electron acceptor. Okay. And this is located where? So, it is located within the inner membrane. Okay, within the inner membrane of what?
Mitochondria.
Very good. So, inner membrane of mitochondria. This much is clear?
Yes.
All right. Now, moving on to the step number 3. Later on, this ubiquinone, apart from receiving the electrons, it also receives the reducing equivalence from the FADH2, or imagine it receiving the equivalence from the FATH2, that is actually generated during oxidation of succinate. So you remember that we had FADH2 being formed from FAD
Mm.
plus. So at that time also certain electrons were released. So from the FADH2, the electrons are being sent to the ubiquinol, and then from the NADH2, the electrons are being sent to the ubiquinol. So it's receiving electrons from both of them.
Okay.
Okay. So we'll write that ubiquinol also receives the reducing equivalence.
What is equivalence?
I'll tell you. Via FADH2. And you already know the location where FADH2 is being needed in the citric acid cycle, isn't it?
Yes.
Okay, very good. So see, reducing equivalence, these are basically the like electrons only. You just remember them as electrons only. Okay. So the electrons from the NADH and from the FADH2 are coming to the ubiquinol. Okay, because here I've written ubiquinol also receives. So earlier it was receiving from NADH2, right? But now it is also receiving via the FADH2. And then let us write where it is generated. So that is generated. What happened?
You have to write NADH2 also, right? Because electrons from that and FADH2 come to the.
Yeah, so we have written, right?
But we didn't write that.
See, the electrons from the NADH produce, they are being oxidized. And these electrons are then transferred to the ubiquinol. So transferred from NADH2 only.
Okay, okay.
Okay. And they are transferred to ubiquinol. And along with that, the ubiquinol also receives the electrons or reducing equivalents from FADH2, which are generated during oxidation of succinate. You remember this? I'll tell you where. Wait. Look here. This part where the FADH2 is being synthesized. It is coming from succinic acid only, right? And during the formation, like all the steps going towards the this oxaloacetic acid formation, isn't it? So here the FADH2 is synthesized. This is what we're talking about. Okay, so the electrons that are produced during this, they are also transferred to the ubiquinol. Getting my point?
Yeah.
Any doubts up till now?
If we would have done this chapter in the beginning, not this chapter, but like plant physiology,
Hmm.
then I would have said that bio is hard.
Then you would have said bio is hard.
Mm.
Yeah, see, this particular chapter is sort of like a tricky chapter. I understand that why, because first of all, it has so many compounds, new
There's
compounds
so
that
many enzymes and new
yeah,
words.
yeah, I know that. But this does not make bio hard. It's just like one part which is tough. Like
Yeah.
not
Yeah,
everything can
now I
be
get that.
easy.
But
Hmm.
my friends in
Hmm.
the beginning of 11th, they did this first, plant physiology. So they found bio so hard. They were like, oh my god, I hate bio and all.
So it's really good that I warmed you people up with nice chapters,
Yeah.
and then I'm showing you the bad side, the dark side.
Yeah.
It's good. It was a good trick that I did, even though that was purely unintentional. But
Yeah.
anyway, it served the purpose. All right. So, okay, basically, let us keep it till these three steps only. Okay, let us revise yet again. So, first of all, what happens is that the electrons from the NADH that are produced in the mitochondrial matrix during which process? So, during citric acid cycle, Now, these electrons are oxidized by the NADH dehydrogenase. This is complex number 1. Later on, these electrons only, they are transferred from the NADH to the ubiquinone. Okay, this is located where ubiquinone is present in the inner membrane of the mitochondria. Now, this ubiquinone, along with the electrons from NADH, also receives the electrons from the FADH too. And where is it synthesized? So it is generated during the oxidation of succinate in the TCA. Now, see, another thing which I think over here is actually making the whole thing complicated is because look at these sentences. They're so huge. These sentences are like connected, like electrons from the NADH produced in the mitochondrial matrix during the process of citric cycle. Now, those electrons, we're not talking about citric acid cycle. We're not talking about NADH. Rather, we're talking about the electrons. So I see where it gets confusing because the sentences are very, very long. And I don't think that you people will be able to remember this exact sentence during your examination.
I will write it
Is
in
it?
another way.
Hmm.
I will write it like
Hmm.
NADH dehydrogenase oxidizes. No, wait.
Yeah, that's good. That's good. See, NADH dehydrogenase, complex 1, oxidizes the electrons which are
Yeah,
produced from NADH in the mitochondrial
yeah,
matrix during citric acid cycle. This is
yeah.
better, right?
That's what. That's what.
Hmm. So you can write it in both ways, but the answer is right. Like the concept should be this only.
Yeah.
Okay, so this is like the 3 steps that we studied about the ETS, and later on we'll have 3 more steps. Okay, so 6 steps are there, and then this topic will be over. I'm so excited for this chapter to get
For real.
over it. So yes, now tell me if you have any doubts.
No. I
No
have to
doubts?
do the test now.
So I would advise you people to read your NCRT because that's the most important thing to do.
Is
And
it
then
easy? Test?
the test, yeah, it's nice,
Yeah.
beautiful. If you have paid attention. If see, to be very honest, if your preparation for the test is good, then the test is going to be good, really good, because the questions are very nice. All right. So yes, so pay attention. Also, yeah. While you're solving the questions, pay attention to what the questions are asking, and write the answer according to the marks that it requires. Okay.
Okay.
All right. So then that is it for today's class, and I'll meet you on Monday.
You said it on in the last.
Yeah. But maybe okay, before saying bye, I should show my face and then I should say bye. And yeah. Also, like the reason why sometimes I switch off the camera during these type of classes, like since the teaching pattern has changed. So like listen, when I write, okay, I go like this. Because I have this habit of being very close to my notebook while I'm writing. So right now my screen is over here, so I'm like getting very close to my screen, and it looks obnoxious on the window, like the screen. So that is why
That's
I don't
okay.
want to be up in your face. So I switch off the camera while I'm writing. Okay.
Okay.
So you people should know that, okay, I don't switch it off because I'm lazy or something. I'm properly dressed for the switching on and everything. But it's just that I write so obnoxiously. Like you might have noticed a sentence like this. And then I'm like
It's
this. And
it's
then I'm up.
okay.
It's weird. So that
No.
is all right. Now listen, it's very important. You are going to give a test, right? After
Yeah.
that, you take out your